Okay, so I think we kind of finished off last time starting to talk about carbohydrates. Um, so big ticket, big topic items here. You should understand what the functions of carbohydrates are. Short-term energy storage, they're converted to glucose quickly, and glucose is used to make ATP or energy in your body. ATP is the body's energy currency, and all of your body cells need ATP to do every sort of function. So we get glucose from carbohydrates, and that's why carbohydrates are important, and brain cells also require glucose. Characteristics of lipids then, so we must have ended with lipids here, I think. Either way, it'll be a review. Lipids will contain carbon, hydrogen, and oxygen. That's what CH and O stand for. They contain a lower proportion of oxygen to carbon. They're insoluble in water. We did talk about this. That just means that lipids, all of your fats, your oils, your cholesterol, triglycerides, and phospholipids, they cannot dissolve in water. And we talked last time, you can do a little science experiment, take vegetable oil and water and try to mix them together, and they will not mix together. And that's why we call lipids, they're insoluble. Functions of lipids, they are more long-term energy storage, they insulate against heat loss, they protect your organs, and cholesterol, which is a type of lipid, is part of your cell membrane structure. And the next chapter, we'll talk about how the cell membrane looks and how lipids are important in that. Lipids can be saturated or unsaturated. Saturated just means every bond between carbon atoms is a single bond. That just means one kind of sharing of electrons is bonding two carbon atoms. And examples of saturated lipids are beef, pork, whole milk, cheese, and eggs. And unsaturated lipids, you have one or more double bonds between carbons. And unsaturated lipids are olive oil, fish oil, and sunflower oil. So which are healthier, saturated fat, saturated lipids, or unsaturated?
I would say saturated.
Unsaturated?
Unsaturated. So if you look at like beef and pork versus olive oil, fish oil, the unsaturated lipids are healthier. And I'm going to show you how. Oh, they don't have a good. Here, this is what I wanted to show you. So I'll go back to the previous slide. So we talked about saturated and unsaturated lipids. Unsaturated and saturated have to do with the bonding, and they're also called fatty acids. Saturated fatty acids, do you see how there's only single bonds between carbon atoms? Because there's only single bonds between carbon atoms, you see this linear pattern of this fatty acid. And because it's completely linear, saturated fatty acids can easily be stacked up on top of each other. And when they stack up on top of each other, they condense and they get held together really tightly. So saturated fatty acids become very saturated, and it's harder for your body to break down saturated fat because of their molecular structure, no double bonds. This linear structure of a fatty acid, because it can stack up on top of each other, it's just harder for your body to break down. And that's why whole milk, pork, beef, that's unhealthier for your body because it's harder for your body to digest them. Versus unsaturated fat, do you guys see these double bonds in here? In molecular chemistry, a double bond kind of causes the molecule to bend. And because of this random bending, unsaturated fats cannot easily stack up parallel on each other. So they're not saturated, they're kind of all over the place, and they're more loosely put together. So because of that, your body can break them down more easily. So that's why unsaturated fat, olive oil, fish oil, avocados, they're unsaturated fat. That's good fat for your body because your body can break it down more easily and get the glucose that it needs out of it for energy. So that's kind of an interesting point, I think. Any questions on that? Okay. So this is triglycerides, are just another type of lipid, and they're called triglycerides because there's 3 groups of a 6-carbon glucose attached to them. So these are triglyceride molecules. And fatty acids are the piece in purple, and glycerol is the piece in pink. And if we combine them, we get a triglyceride. But anyway, triglycerides are just another type of lipid. Fatty acids can be saturated or unsaturated. Phospholipids are unique to lipids. They have a head, this little circle region, and then a tail. And the cool thing about phospholipids is that the head part is called polar or hydrophilic. That means it loves water because water is a polar molecule. So the head of phospholipids loves water, and the tail of phospholipids is nonpolar. It's hydrophobic. That word phobic or phobia means afraid of. So the tails are water fearing. They don't love water. And we'll look at phospholipids when we get to the cell membrane in the next chapter. Then proteins contain carbon, hydrogen, oxygen. So the same as the previous structures, but they also contain nitrogen. Amino acids are the building blocks of proteins. And there's 20 different naturally occurring amino acids. So these 20 different amino acids will form in different ways to create different proteins. Amino acids contain an amine group, which is made up of nitrogen and hydrogen, and some sort of carboxyl group, which is just carbon and oxygen. Amino acids are not stored, so a daily supply is required of amino acids. Here's amino acid structure and bonding. These are two examples of amino acids, alanine and glycine, and it's just showing how they are bound together through a bond. And if you put several amino acids together, you get a protein. So big topic item, amino acids, you put several amino acids together and you make different proteins. And there's 20 different amino acids that are naturally occurring. The linear sequence of amino acids in a protein, this is just showing a long chain of amino acids. So you can see here a protein consists of a chain of different amino acids, and each amino acid is just a different color. So the blue might be the same type. So this is just a protein. This is one protein, and it's made up of different kinds of amino acids. Proteins will also fold. So proteins will be linear in a line, and then they'll fold on top of each other. And we call that folding kind of a 3D or three-dimensional representation of the amino acid chain. The amino acids in DNA, for example, can also be bound together by hydrogen bonds. And those hydrogen bonds will bind those, the chains together, and that'll cause the amino acid chain to become folded or also coiled, like in DNA, which is carries all of our genetic material. An entire protein has a complex three-dimensional shape, and that's what's shown here. So there's a linear aspect to it, then it'll fold on top of each other, and then could even coil itself around it. So this kind of shape that you see over my cursor, this is what a protein actually looks like if we were to look at it in a very high objective. Functions of proteins. Your proteins are used for everything in your body. They are used to make your skin, hair, nails, muscles. They're part of the hemoglobin molecule, and hemoglobin is a molecule that carries oxygen in your red blood cells. Proteins will act as enzymes, and enzymes are helpful in making chemical reactions go faster. Proteins will help with your immune system, so keeping your body healthy and also fighting off disease. Proteins help in muscle contractions. So actin and myosin are two types of proteins in muscles. They slide past each other to shorten your muscles, and that's what causes your muscles to contract and pull your body parts around. And they're also part of the cell membrane that we'll get to in the next chapter. Protein denaturization. What does denaturing mean? In fitness, you hear the word a complete protein. What does that mean? That's a good question, Brian. I think that might be something not made up by the fitness community, but I'm not exactly sure what a complete protein would be, but I'm assuming it's one that's healthier for you in some way. Good question about that. I'm not sure about that one. Protein denaturation occurs when the hydrogen bonds that maintain the shape of a protein are broken and the protein becomes non-functional. So if we denature a protein, that just means we kill it off, and when it's unable to perform its job, then it becomes non-functional. And proteins can be denatured by high temperatures or improper pH levels. So too acidic, too basic, anything that would be out of the normal pH for it to function, that would cause it to be denatured. And denaturing would just break the bonds that hold the protein together, and it would become non-functional. So that's what denaturing means. Enzymes are what we call organic catalysts that increase the rate of biochemical reactions. They'll proceed without the enzyme being permanently changed. So enzymes will stay the same throughout the course of a chemical reaction. They'll just make their reaction go more quickly. And enzymes work by lowering the energy of activation. And the easiest way to understand the idea of enzymes lowering the rate of activation. So remember, in a chemical equation, you have products and you have reactants. So you have something you start with, the reactants, and then you have something you end with, product. And an enzyme will come in and lower the activation energy. So if you see this big kind of up and down bump in this graph, that's the activation energy. Think of it like a hump or a bump that the chemical equation needs to get over to proceed to get products at the end. All the enzyme does is it lowers the energy needed to cross over that hump. So enzymes will lower the activation energy, so it's easier for your reactants to get up and down that hill if the hump or bump is just lower. So that's what enzymes do. They act as catalysts. They are proteins, and they just lower this level of activation energy, meaning it's easier for reactants to get over that hump and to form products at the end. So that is kind of a representation of what enzymes do. Thanks for answering the question. I didn't actually know that. So a complete protein is a protein source that contains a proportion of all 9 amino acids. Yeah, I wonder what 9 amino acids. Yeah, there are 9 amino acids that are needed in the diet that your body doesn't have. I don't know what they are, but if you guys want to expand on that in the chat, that's interesting. Essential, yes. Great chat questions. Enzymes action. So if we look at our enzyme, they work as like a lock and key model. So the enzymes will take 2 molecules, which would be your reactants in a chemical equation. It'll bind them both, put them together, reform them, whatever needs to happen, and then they'll spit out a new molecule, a product. So again, an enzyme just helps a reaction proceed more quickly and more easily by lowering the activation energy required. Nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And examples of a nucleic acid that you guys should be familiar with is DNA and RNA, and there are little differences between the two. Nucleotides are the building blocks of nucleic acids, and nucleotides are composed of a nitrogen base, a phosphate group, and a 5-carbon sugar. So this is the structure of DNA, and we'll just briefly kind of talk through this. The building blocks of nucleic acids are nucleotides, which are seen here, this bracket. So they contain this phosphate group, some sort of sugar, and the sugar in DNA is called deoxyribose. That's why it's called DNA for deoxyribose. RNA, its sugar is just a ribose sugar, so that's one difference. And then it's made up of a nitrogen base. And in DNA, there's 4 different types of nitrogenous bases. T stands for thymine, A stands for adenine, G stands for guanine, and C stands for cytosine. And these nitrogenous bases will be bound together with hydrogen bonds, which again, hydrogen bonds are the weakest bond. And they're bound, when they bind together, they will form this kind of helical or twisted pattern to DNA. These nitrogenous bases have to bind together in pairs. So thymine only pairs with adenine, and guanine will only always pair with cytosine. So that's what this picture is showing down here, just that these base pairs will always pair together, C with G and T with A. Adenosine triphosphate, again, ATP is your body's energy currency. So it's like the money that your body needs to pay for energy and do all the sort of processes in your body. It's an especially important organic molecule found in all living organisms. It consists of adenosine, which is a ribose sugar with an adenine base group, and then 3 phosphate groups that are bonded to it. That's why we call it adenosine triphosphate. It's an adenosine group, and then triphosphate means the 3 phosphate groups that are bound to it. It's called the energy currency of cells because it's capable of both storing and providing energy. It stores energy in its bonds to its 3 phosphate groups, and it provides energy when those bonds are broken. So here's a structure of ATP. Here's the adenosine group, and then its 3 phosphate groups. Adenosine diphosphate, you might hear of, that's ADP, that has 2 phosphate groups, and adenosine triphosphate has just 3 phosphate groups. So that's what these brackets show, just the addition or subtraction of a 3rd phosphate group. So that's ATP, and that takes us through the end of chemistry, which you guys are probably so excited to be done with. Any questions on the end of this chapter before we start? I just want to start chapter 3, which is on the cell, because if you've looked at it, there's 100 slides. Any questions on this? Okay, and again, feel free to shoot a question to me privately or publicly in the chat if you do have a question. And I'll pull up the next PowerPoint. So give you time to think of some questions here.
What are the chapters that the first exam is on?
The first exam are on chapters 1 to 4.
Okay, thank you.
Yep. Good question. And the lab exam will be on exercises 1 to 5. So in Canvas, I put all the files up at once. But in Canvas, you'll see there's lecture and lecture, just the first 4 chapters, and lab would be the first 5 exercises. All right, so let's start chapter 3. Hopefully chapter 3 is a little easier to understand because it's just learning about a cell. And I'm hoping that this might be more, not a complete review, but at least you might have heard of some of these cell structures and their functions before. The cell structure itself, so we're talking about every living thing is made up of cells, and every cell is a little different depending on what its purpose is. But most cells have organelles, which are tiny organs. They're specialized structures in cells that perform very specific functions, and we'll go over some of those organelles. The cytoplasm is a jelly-like substance inside the cell that holds all the organelles in place. And the cell membrane, also known as a plasma membrane, it encloses the cytoplasm and it keeps everything in the cell. It also controls substances that go into the cell or go out of the cell. Yes, Ryan, hydrogen bonds are weak and they would fire easily break down DNA. Fire. I would assume fire would, but the weakness of the hydrogen bonds in DNA especially is important because DNA needs to be replicated, so it needs to make copies of itself in order for cells to divide and pass on their genetic material to the next cells. So that's why DNA is made of those weak hydrogen bonds between bases, because when it uncoils, it can replicate itself. So that's why those hydrogen bonds are weak. So I'm assuming fire could easily break them down or anything else. Oh, no, that's okay. I'm obsessed with crime shows too. That's not totally fine. All right, so cell structures. Cell membrane keeps everything in place. It's kind of the wall, exterior wall of the cell. So here's a look at a cell. I think we go over a cell in lab or one of your exercises. You should know how to label some of these structures, but I think your lab book and those virtual lab links will take you through some of this. So in a cell, here is the cell membrane or the plasma membrane. It surrounds the cell. The nucleus is like the brain of the cell, and the nucleus has its own kind of wall to it, which is what we call the nuclear envelope, and the nucleolus is the center part of the nucleus. Then we have different structures or different organelles in the cell. Here's the rough endoplasmic reticulum is kind of the part that surrounds the nucleus. It looks like flattened tubes that surround the nucleus, and it's called rough because it has ribosomes on it. You also have smooth endoplasmic reticulum, which is not shown here, which is okay. Well, yeah, here it is. The smooth endoplasmic reticulum will be a continuation of the rough endoplasmic reticulum, but the smooth does not have ribosomes on it. Then you have a Golgi apparatus is here. Mitochondria are these kind of weird structures that look like there's another space within them. That's mitochondria. You have lysosomes. You have different vesicles taking things into the cell, taking things out of the cell. You can have ribosomes that are what we call free floating, that are just floating in the cytoplasm. You have centrioles. So a lot of stuff goes on inside the cell. And we'll kind of go through them now. Let's talk overview though, the functions of a cell. It's the smallest unit of life. It's able to perform cell metabolism and use energy. That means it takes in glucose and breaks it down for energy use. It can synthesize new molecules. It communicates with other cells, and it also reproduces and spreads, or by reproduction, it'll pass along its inheritance or its genes in the form of DNA. So that's what inheritance means. It passes on its genetic material. The cell membrane or plasma membrane is the outermost component of a cell. It forms a boundary between anything inside and outside. Materials inside the cell, we call them intracellular. So this prefix intra means inside the cell, and anything outside the cell would be extracellular. It acts as a selective barrier. So that means this cell membrane is a barrier between everything inside and outside of the cell, but everything doesn't just rush into the cell. It's selective in what it allows to come in, and then it's very selective with what it pushes out of the cell. The cell membrane structure, we call it a fluid mosaic model. Mosaic means a lot of different pieces are with it, and fluid means all of these different pieces of the structure are kind of constantly moving within it. So that's what fluid mosaic means. The membrane is made up of phospholipids, cholesterol, proteins, and carbohydrates. Phospholipids form a bilayer or a double layer, and I'll show you a picture of what that looks like. And the phospholipids contain two regions, a polar and a non-polar region. So let's get through the phospholipid structure, and then I'll show you a picture of what this is. From what we just talked about, if you remember, I told you a phospholipid has that polar head that loves water and a non-polar tail that is afraid of water. So the polar region will be exposed to the water on the outside of the cell and the inside of the cell, and the non-polar region will be facing the interior of the membrane. And this is what I mean by that. So all of these blue little circles, that's the head part of the phospholipid. And again, the head part loves water. It's hydrophilic. So it will be in contact with water on the exterior side of the cell and also on the interior side of the cell. So again, this is a structure of the cell membrane. We call it the phospholipid bilayer because there's two layers of these phospholipids. The head part of those phospholipids touches the water, either exteriorly or interiorly. And the tail region, do you guys see how the tail regions all touch each other? That's because the tails are water-fearing. So in the interior of this cell membrane, there's no water there. It's a non-polar region. These tails are touching each other because they're water-fearing. They're non-polar. They don't want to touch water. So they all line up so that the tails touch each other and the heads will be facing kind of outward. Hopefully that kind of makes sense. Within this cell membrane, we call it fluid mosaic because there's a lot of different pieces in it. You see protein membrane channels, which allow things to go in and out of the cell. You see receptor molecules, which will bind to just things that the cell needs to recognize. There's carbohydrate chains, there's cholesterol embedded. So you'll see just different parts of the cell membrane. We call it fluid mosaic because there's a lot of different pieces and they're in constant motion. Questions about the cell membrane? Okay. Movement through the cell membrane, it has what we call selective permeability, which only allows certain substances to go into and out of it. Substances like enzymes, glycogen, so enzymes help to make reactions go faster. Glycogen stores glucose, and potassium is an ion that your cells need. They're found in higher concentrations inside the cell, and substances like sodium, calcium, and chloride are found in higher concentrations outside of the cell. Cell membrane passage. So how do things go through the cell membrane to get into the cell where different organelles might need them? Oxygen and carbon dioxide can pass directly through the cell membrane's phospholipid bilayer. So oxygen and carbon dioxide can go freely into and out of cells. And that's really important because your cells will need oxygen to make energy, which is ATP, and carbon dioxide is like a waste product of all of your cells when it makes energy. So it's important that oxygen and carbon dioxide can get into and out of your cells really easily. Some substances must pass through what we call protein channels, and this is a look at a protein channel. And some substances will pass through those channels, such as sodium. And the route or way of transport through the membrane will depend on the size, shape, and the charge of the substance. So if things are too large or if they have a charge, they might not be able to pass through. And depending on their shape, they might not be able to get through. Some substances will require a carrier molecule. So think of it just like someone who's helping a substance get across, such as glucose. Some substances require a vesicular transport. That means they'll come across the cell membrane in a vesicle or like a container. Think of a vesicle like a container. That vesicle or container must fuse with the cell membrane, so it kind of will join the cell membrane and then be spit out on the other side, depending on which way it's going. Really simply put, we call these transport processes through the cell membrane, they're either passive or active. And a passive membrane transport does not require the cell to expend any energy. So passive membrane transport just means that things can come freely into or out of the cell passively without any extra energy. Active membrane transport does require the cell to expend energy. So active membrane transport will need ATP to get anything into or out of the cell. Passive membrane transport mechanisms, which we'll go over, are diffusion, osmosis, and facilitated diffusion. So those three bolded terms do not require any energy. And active membrane transport mechanisms include active transport, secondary active transport, and then endo and exocytosis. So again, big picture things, we're talking about actively and passively getting things into the cell or out of the cell through the membrane. And passive mechanisms do not require energy. Active mechanisms do require energy. So then we'll go through some of the passive mechanisms first, and we'll talk about diffusion. And before I start to read off of a slide, because this is no fun, my biggest example for diffusion is, well, I have two examples. One that I love is coffee, and I like cream in my coffee. So I have a cup of black coffee, and I put a splash of creamer in it. The creamer will diffuse to all parts of my coffee cup until an equal concentration is reached between coffee and creamer. So that's a diffusion. Diffusion just means that a substance moves down a concentration gradient from an area of high concentration. When I first put my coffee creamer in, where that creamer goes, that's a high concentrated creamer area. And then it'll move to an area of low concentration. The creamer will be actively dispersed. I don't know if you guys like coffee with creamer. Another example of diffusion, if we were in a classroom and I would spray perfume or cologne in the front of the classroom, that area would have the highest concentration of that perfume. Through diffusion, that perfume would disperse through all parts of the room until there was an equal concentration of perfume, maybe to a point where you wouldn't even smell it anymore. So that's just what diffusion is. Diffusion is a movement of a substance from an area of high to low concentration, and that's called a concentration gradient. A solution is composed of two parts, the solutes and the solvent. So in my cup of coffee, the coffee was the solvent and the creamer was the solute. And when they're mixed together, they form a solution. Solutes will be substances dissolved in the predominant liquid or gas, which is called the solvent, and the solutes will move from an area of high concentration to low concentration. And that's just what diffusion is. The concentration gradient is that difference in concentration of a solute and a solvent between 2 points divided by the distance between the 2 points. What that just means is a concentration gradient is said to be steeper when the concentration difference is larger or the distance is smaller. So if we say the concentration gradient is very steep, that means there's just a higher concentration of the beginning solute particles. Let's see if we have a picture of this. So if we have a beaker of distilled water, which would be the solvent, and we drop in a salt crystal, which is green, the salt crystal is our solute, and the green salt crystal will disperse. So through diffusion, the water will kind of mix up that salt, and through diffusion, the salt will be equally distributed to all aspects of that beaker container. When we mix the solute and the solvent together, again, the solute is the salt crystal and the solvent is water. When we mix them both together, we get a solution of equilibrium, and that just means equal concentration of salt and water. Questions on diffusion? Leak and gated channels. These are channels within the membrane. Lipid-soluble substances can diffuse directly through the phospholipid bilayer. And water-soluble substances like ions can only diffuse across the cell membrane by passing through a membrane channel. And a membrane channel, a channel is just kind of a hole in the membrane that allows water-soluble substances to get through. There's two types of channels. They can be leak or gated. Leaking channels, just like a leaky hose or a leaky faucet, this will be constantly open to allow ions to pass through. And gated channels, like the name says, will limit the movement of ions across the membrane, like with a gate-like mechanism by opening or closing the gate. So here's diffusion through a cell membrane. Here's the membrane channel, which is just an opening through the cell membrane. Non-lipid soluble molecules or ions can go through there. Non-lipid soluble molecules sometimes are unable to go through. Lipid soluble molecules are able to pass directly through because of the phospholipid bilayer. Only lipid soluble molecules will be able to get through. Oh, no problem, Ocean. Thanks for putting your attendance. The concentration gradient is shown here. If you look specifically at these red molecules, there's a greater concentration of red molecules outside the cell. So when this membrane channel opens, whether it's a gated or leaky channel, These red molecules will pass into the cell passively because they'll always travel from an area where they're more concentrated to an area where they're less concentrated. Leak and gated membrane channels. This is just showing a picture. Let's see, a gated channel, you can see here there's a close and open functioning to a gated channel. And a leaky channel is constantly open and things will just leak in and out of it. So that's just the difference between a leak channel, it's always open, and a gated channel will have a gate mechanism. And again, these are channels that help things pass through that phospholipid bilayer that are ions, or they are non-lipid soluble. Osmosis is still passive. So osmosis is another transport mechanism. And osmosis, really simply put, is the diffusion of water, which is always a solvent, across a selectively permeable membrane from a region of high water concentration to a region of low water concentration. So osmosis, when we talked about diffusion, we talked about solute particles being diffused. Osmosis is the specific term for the diffusion of water. Osmosis will exert a pressure, it's what we call osmotic pressure, which is the force required to prevent movement of water across a cell membrane. A sodium channel only opens to what specifically? If it's a sodium channel, it only allows sodium to pass through it. So a lot of channels are named for the substance that will pass through them. You'll have sodium channels, potassium channels. Some will just be leaky channels and anything can get through if it's small enough. But a sodium channel would open only up to sodium. Okay, so we're still in osmosis, osmotic pressure in the cell. Osmotic pressure, again, helps osmosis to occur. This depends on the difference of solution concentrations inside a cell relative to the outside of the cell. And this is a part of the PowerPoint that I encourage you to listen to because I know there's at least one test question on what a hypotonic, isotonic, and hypertonic solutions are. A cell can be placed in solutions that are either hypotonic, isotonic, or hypertonic compared to the cell cytoplasm. Hypotonic, the prefix hypo means less concentrated than the cell. The prefix iso means equal. There'd be an equal concentration between the inside of the cell and the solution. And a hypertonic solution would be more concentrated. It will have a higher concentration of a substance compared to the inside of the cell. So hypotonic, isotonic, and hypertonic. A hypotonic solution has a lower concentration of solutes and it has more water in it. And that's relative to the inside, the cytoplasm of the cell. The solution has less tone or osmotic pressure than the cell, and water will always move by osmosis into the cell, causing it to swell. And I'm going to show you a picture after we get through these three definitions of what I mean by this. But again, water will always move into an area of higher solute concentration. And if a cell is placed in a hypotonic solution, the cell will swell and it could rupture. And we call that, if a cell actually ruptures or breaks, we call that a lysis. And a lysis means to split open. An isotonic solution, if a cell is put into an isotonic solution, it will have the same concentration of solute inside and outside of the cell. So water can still move freely into and out of that cell, but the cell will neither shrink nor swell. This is what we want our cells in our body to be placed in, an isotonic solution, because this will not cause your cells to split apart or decrease in size. And then a hypertonic solution, the cytoplasm of a cell is put into a hypertonic solution, and the hypertonic solution will have the greater concentration of solute, and the cell will have the lower solute concentration. Water will move by osmosis from the cell into the hypertonic solution, and when water leaves the cell will shrink. And we call, anytime that a cell shrinks in size, we call that crenation. So let's talk a little bit about what osmosis is, and then we're going to review what these hypotonic, isotonic, and hypertonic solutions mean. Varicose veins, Samantha. Sorry, I'm going fast, Larry. Yes, these slides are posted, and I also put the recorded lecture online. Yes. Okay, so osmosis. Osmosis, again, is the diffusion of water. Oh, I don't like this picture of it, but we'll talk about this one anyway. Okay, so osmosis, if you look at this beaker where my cursor is, this beaker is filled with a salt solution. So there's a lot of salt in this kind of beaker. There's a selectively permeable membrane, which is holding the salt in this solution. So this is a salt solution of 3%, and there's a selectively permeable membrane covering it up so the salt isn't able to get through. If we put this kind of beaker container into just water, just distilled water, as we put it into distilled water, we'll see what osmosis does. So through osmosis, water will travel from an area of high water concentration to an area of low water concentration. So if we zoom in on what's happening here at this picture where my cursor is, which way is water going to travel? And again, water wants to travel to try to equalize the concentrations on both sides of this membrane. So water will always travel into an area of higher solute concentration. So water is going to travel inside this salt solution. Because this membrane is selectively permeable, that just means that water can get through it, but salt cannot get out of it because the salt crystals are too big to leave. So osmosis is water moving into that higher solute concentration. When it does that, when water moves into the salt concentration, it will change this level of solution, and you can see how the salt solution is rising and traveling up as because water is moving into it. Does that make sense? No questions? Okay. So how does this relate to your body cells? So let's see, one more. Okay, thanks. So why is this important? These are pictures of your red blood cells. If you're ever given a blood transfusion or a saline solution, I just got one when I had my baby and was given an epidural. So if you're ever in the hospital and they're giving you a saline solution, the concentration of ions in that saline solution has to be isotonic to your red blood cells, or your red blood cells could split apart or they could shrink. And this is through osmosis. So if we take a red blood cell, and these are your red blood cells, they make up your blood. If we put red blood cells into a hypotonic solution, we are putting them into a solution that is less concentrated than your red blood cells. So we're basically putting red blood cells, let's say, into basically just distilled water. So if we put red blood cells into a hypotonic solution, meaning a solution that's less concentrated than what is inside your red blood cells, water is going to move from an area of high water concentration where there's more water into an area of less concentration water. So water travels into your cells if we put a red blood cell into a hypotonic solution. This will cause your cell to swell or increase in size, and if it swells too much, your cell could actually split apart. So this is why it's so important to get the correct concentration of a saline solution or an IV that you're giving someone in respect to their red blood cells, because you don't want to put their red blood cells into a hypotonic solution, water rushes into your red blood cells, and your cells will swell or split apart. So that's a hypotonic solution. If we put our red blood cells into an isotonic solution, we're putting them into a solution where there's an equal concentration of solutes, whatever they may be, inside the cell and outside the cell. So water will freely move into and out of the cell, but not only in one direction. So that's an isotonic solution. And then a hypertonic solution, if we put your red blood cells into a hypertonic solution, what does hypertonic mean? Where's the greater concentration of solutes? In the solution or in the cell?
So the solution is oversaturated.
The solution is oversaturated, yes. So we're putting a cell into a hypertonic solution, extremely concentrated solution of solutes. When that happens through osmosis, water, again, always travels into that higher concentrated solution. So water will leave the cell. Another way to remember which way water always travels, if you can remember the phrase solutes suck and solutes suck water. So remember that solutes will always suck the water into them. That's true for a hypertonic solution. The solution is more concentrated, so the solutes in the hypertonic solution will suck the water into them. Water leaves the cell and the cell shrivels up. If we put a cell into a hypotonic solution, there's more solutes in the cell this time. So the solutes in the cell will suck water, and water will enter the cell, and the cell will enlarge. Question?
So this is,
yeah.
Sorry. So this is basically, I guess, a relatable way of thinking of it is dehydration, and then basically being bloated with salt water and etc.
Yeah. So why is it bad to drink salt water? What happens to your body?
Your body retains it, so you get kind of like a bloat.
That's true. And yeah, and specifically when your cells are put into salt water, when your cells are placed into a hypertonic solution, all your cells, the water inside your cells leaves and exits your cells. So never drink the salt water, because when you drink salt water, you're filling up your body with a hypertonic solution. So all of the cells in your body, water will leave all of your cells and osmose or diffuse into that salt water. And that's what dehydration is as well. But specifically, yeah, if you're ever in need of water and you're stranded at sea, don't drink the salt water. And this is why. You guys knew that, but now you know why. This happens with fish, whether you have a saltwater or a freshwater fish. Fish also have other mechanisms to handle their different concentrations of salt versus fresh. But simply put, this same thing will happen if you take a saltwater fish and put it into a freshwater lake and vice versa. You'll mess with their cells too. Okay. A couple more slides here. Let's just try to get through facilitated diffusion, which is the end of the passive transport. A carrier-mediated transport. So we're still talking about passive transport. We're still talking about a cell membrane. I know you're probably lost right now, but we're talking about cells in this chapter. The membrane is the exterior side, and we're talking about how things go into and out of the cell. We're covering all the passive processes first, and passive processes do not require energy. A carrier-mediated transport has to do with a carrier molecule that helps things into or out of the cell through diffusion. Some water-soluble, electrically charged, or too large particles cannot enter or leave the cell. These substances would be amino acids, glucose, and some polar molecules. So you'll have a carrier molecule, which are proteins within the cell membrane that just help get that molecule into and out of the cell. Carrier-mediated transport can be passive, which is facilitated diffusion, or it could be active, which requires ATP. So facilitated diffusion does not require energy. Active transport would require ATP for a transport of these molecules. Facilitated diffusion is just a carrier-mediated, so it uses a carrier channel protein that moves things across the membrane from an area of high concentration to an area of low concentration. Because it moves from a high to low concentration gradient, which is diffusion, no energy is required. And this is just a look at this little carrier molecule. You can see the channel that goes through it. You can see glucose entering it, but it just needs the help of a carrier molecule to get it through this lipid bilayer. Because glucose can't diffuse directly through the lipid bilayer, it has to use a carrier molecule to get through it. And that's facilitated diffusion. And I think I'm going to stop there for questions. And we'll continue. I know we have a lot to get through on Thursday, but I'm going to just stop here for questions. And you can ask me questions now. Let me stop the recording.